So hey guys, welcome back to a new video. And in the last video we looked upon the general structure of virus. So in today's video looking into the life cycles of the virus, and we have got two types of life cycles. One is the lactic cycle and the other is lysogenic cycle. And in today's video looking into the lactic cycle, right? uh, Let me zoom a bit. Okay, so we will be discussing about the lactic cycle, and we'll be looking into the diagrams to understand it. I'll be writing a less theory, but you will be get cleared about the actual concept. So first of all, let me tell you that this is a life cycle that mainly occurs in event phases, and in this process the host cell gets destroyed. What do I mean by that? Like, lactic cycle is a kind of cycle which mainly occurs in event phases. Like event phases, let me tell you about it. What does that mean? We've got different type of type 1, type 2, type 3, type 4, type 5, type 6. Phases means bacteria. So bacteria, so different type of bacteria have got different life process. Some have got lactic cycle, some have got lysogenic. So even phases, like we can say that this means that T2, T2, T4, T6 we have also got. So in these type of viruses or phages, this gets happened. And uh, in this process we the host cells get destroyed. You need to remember this point, because the lysogenic cycle is kind of cycle in which the host cell does not get destroyed, but in the lactic cycle it does. So moving forward into it, in the main concept, so let me draw the bacterial cell or the host cell. Okay, that is, we're looking into the lactic cycle of bacteriophage, and bacteriophage is the bacteria eater. We looked into the general structure of bacteriophage in the last video, and uh, we'll be looking into it. And what we'll be doing first is that, let me draw a bacterial cell or a host cell. This is the host cell, and this is the bacterial DNA. Like over here, this is the bacterial DNA. uh, Let me bacterial DNA. Okay, this is the bacterial DNA, and this is the bacteria. And what happened is that uh, virus comes, and this gets, okay, so this, the very first step is attachment. In other words, we can say this is also called adsorption. This is, remember, not absorption, this is adsorption. And this, the tail fibers get attached to it. Like, the tail fibers attached to the cell membrane of the bacteria or the host cell. This is the very first step. The second step is the, let me show you. This will be like this, going into a, like this, and now the penetration takes place. What happens is that okay, so a kind of enzyme is released over here, this, and this is called the Yes, this is lysozyme. Lysozyme. This is the kind of virus that digests the bacterial cell wall and helps in the penetration of the bacterial DNA into it. So this gets happen like we have got the bacterial DNA. This is over here in the green color, and this is the lysozyme enzyme being released, and this in the result of it, what happened is that uh, this wall gets digested. Okay, wait a minute. So this wall gets digested. What do I mean by this is like this gets digested and this gets opened, and there is a way into it. We have the virus has got a way into it by the penetration, and this is just because of the lysosome and the spikes that are present on the base plate. So they both perform it, and the next step which we have got is the ingestion or the insertion. So what happened is in it is like this is a cell, and okay, wait, we have got a uh, like this, we have got a space from where the cell will get okay. So due to the release of lysozyme enzyme, we have got a contraction in the tail tube of the virus. This is the tail tube, and this is the attachments. Lysosome got released, and we have made a way, but due to the contraction, the release of the lysozyme, there are the contraction in the contractile sheath of this. So due to this contraction, the genetic material of the DNA get released into the bacterial cell. Like this is the tail tail region, and here we have got the you know genetic material being passed in this hollow tube, and we have got uh, contractile shield wrapping it. So what happened is this is the bacterial DNA, and this is the viral DNA over here in the blue color. This is the viral DNA, and this gets enter into it by the insertion or ingestion. And the next phase is the biosynthesis or the replication of the phage. uh, the next step is uh, like this. Okay, so this part over here is now just left behind. This is called ghost, because this is not having any rule now, because this is just an empty portion, and this is not got a use. This will get destroyed, get back into its place, whatever is that. We are in the need of genetic material, and the genetic material or the DNA hijacks the genetic material. Viral DNA hijacks the genetic material like this, and now what happened is that the replications take place, and due to it, what happens is this: the copies are synthesized. Biosynthesis takes place like this takes place in parts, like we've got half parts. Okay, so like now the machinery of the bacteria is being used by the virus, and virus is producing its own copies. This is the tail, this is the head, and these are being produced in the various segments. This this is the uh, what we can say, that this is a master slave relationship. We are also showing this lactic cycle is a master slave relationship. So the reason is simple, like the bacteria is being treated as a slave, and the virus is some kind of master, and it is using the machinery of the bacteria and producing its own copies. And what will happen is next is assembling. We've got assembling, like we have got all the segments. Now these will get stick together, forming a new copies of viruses. And this is how the bacteria survives. Like we've got a proper virus, yes. And this this and the number gets increased time by time. Like uh, this is the replication rate is very high, right? Like we can say that within 25 minutes 200 copies. That is quite high rate. And after a few time the cell gets burst, because you know, there is also a release of a ligase enzyme to brush the cell and digest the bacterial cell wall. But the pressure also get increased, like every 25 minutes the number of the viruses is being increased. So like this happens, uh, this will burst. And the viral segments, the viroids, we've got viroids over here. Proper viroids are the one single particle of viruses. This will get released into the body. This will be going into whatever place you want, and this will be happening. The lactic cycling will be taking place again. And in the result the whole cell is got destroyed, and eventually the whole body get destroyed as well. Like this is destroyed or killed. And this is happening in some kind of in our body. E. coli is also present, and the that is also going for the lactic cycle. And if this got happens, we will be also you know, uh, the bacterial cells are present. The E. coli bacteria is present in our stomachs, intestines. It is helping in the digestion. So this can also happen to it, and we can die because of that. So we are locked into this, and we looked into, you know, let me give an overview. Like we've got this virus, first of all this landing, then we've got the lysozyme. And lysozyme, there's the you know, that makes a whole penetration. And we have got the bacterial DNA being inserted inside. Then we have got the hijacking of that by the viral, and now in this machinery is being used and it produces its own copies. And in the next steps the assembly take place, and as the pressure increase with this rate that is very high, so these got released into the body, and that affects a lot. So I do hope that you got understand and meet you guys in the next video. For that, till that, goodbye.